So, first, I will talk about the fibronectin gene. So, as you know, we have listened in the previous talk that we have about 23,000 genes, more or less, but we still, even if we know the sequence, and the sequence has been known for the last 10, 12 years, more or less, we still don't know which is the function of the gene. We know that the approaches we have seen so far, the high-throughput approaches we have listened in the previous days, they mainly are directed to the study of the gene function by doing a complete gene knockout of the gene. So just by eliminating the function. So, but we have to have in mind that most of the genes, about 80–90% of the genes, undergo alternative splicing, so they produce different isoforms, different proteins that are encoding from a single gene, and in many of the cases, it happens in a tissue-specific manner. There are also alternative promoters and also alternative polynylation signals. About 50% of the genes also have alternative polynylation signals. In some of the cases, the protein that is encoded by the usage of the different signals is different, and obviously the function is different, and it also could happen in a tissue-specific manner. So it is known that some of the genes, for example, this is the microtubule-associated protein tau, in which in the normal case you have a ratio between inclusion or exclusion of this exon, that is exon 10, when there is a point mutation that affects the splicing efficiency through an enhancer splicing, exonic splicer enhancer, then there is a variation in the splicing rate, and this leads to disease. The same happened here in the case of the CFTR gene, in which polymorphic variants of the 3' UTR of the gene lead to mild form of the cystic fibrosis or very severe form of the cystic fibrosis disease. So the idea is how we can determine the function of proteins generated by alternative splicing. And the first thing, we selected a model that is the fibronectin model. And I thank Tito Barale, who is here in the audience, for this selection. He was working in the fibronectin splicing when I arrived to his lab. And we decided to study the function of this exon, the EDA exon. The fibronectin gene is a very important protein. If you do the knockout of the gene, you have embryonic lethality. There are two forms. One is in plasma, and one is present in the extracellular matrix of tissues. This is a very large protein, about 220–250 kilodaltons, and it undergoes alternative splicing in 3 different sites. This exon that is called EDB, this one, EDA, and 3CS by type 3 connecting sequences is the name. So there are, in the different species, there are different number of variants. All 3 variants, they undergo alternative splicing in an independent way. So in humans, we have at least 20 different protein isoforms. So it's a protein that is found in plasma, that is soluble in plasma. It is produced by the liver. The hepatocytes secrete plasma fibronectin that does not contain this exon and this exon. Instead, the form that is normally found in the extracellular matrices of tissues contain variable proportion of these 2 exons. And it has many functions. It has been determined always, in most of the cases in vitro, that it participates in cell addition, in extracellular matrix assembly, embryogenesis, blood clotting, wound healing, and in the oncogenic transformation and other variants. So we decided to study the EDA exon because of different reasons. So one of the reasons is that it is very tightly regulated. So it is, as I mentioned before, it is included in extracellular matrix of tissues, and it is not included in the liver. But regulation of splicing occurs also during aging. At the beginning, in the embryo, it is highly included, as we can see in this slide. So here is the embryo. You can see that this is a PCR, RT-PCR of the fibronectin EDA exon, and it is highly included in the embryo. And as long as we go to older animals, then we can see here that the inclusion decreases. And you can see also that in the liver that is here, you have inclusion in the embryo, and then in the older animals, even after birth, it is not present anymore. And what happens also with the EDA exon is that it is upregulated again when you have proliferating tissues, in the case of wound healing, or in the case of some diseases such as liver fibrosis. The EDA exon also is very conserved along evolution. It has about 87% of amino acid similarity between 22 species, and this is really high. So also the EDA exon has a function. It binds to integrins. I think we have here. And the binding to integrin is supposed to be activated according to the different isoforms that we analyze. In the case in which the EDA is not included, the protein has a closed compact form, which is less active. And because this domain that is called the cell binding domain that contains 3 amino acids, RGD, which are active in the binding to integrins, they are a kind of barrier. They are hidden by the structure of the protein, and when you include the EDA, then the protein gets extended and gets more active, and this domain can be reached by the integrins, and the integrins then have a cascade that activates cellular processes. So, what was the strategy to do so? So this is the splicing site of the EDA. The EDA contains splicing sites as all the exons, but the strength of the splicing site is dependent on the conservation or in the similarity to the consensus site. In the case of the 5' splice site, we have the GT, and then there are 2 other bases that are not conserved and make this splice site weaker. And the same thing happens with the 3' splice site. So what we did was to modify the splicing site. We tested the efficiency first in a plasmid, in a mini-gene construct, in which we included the -1, all the intron, the EDA, and the +1 exon, containing or not the modification in the splicing site or the deletion of the exon. And you can see that in different cell types, the lane 1 corresponds to the wild-type construct. This is transfection into cells and then RT-PCR. This is the wild-type situation in each cell type. And when we transfected the construct containing the mutation, we observed in all cases the full inclusion of the exon. So with this in hand, then we went into the animal. So we constructed by the classical way by gene targeting, as we have seen before. Then we eliminated whatever could disturb the expression of the gene, such as the neomycin selection cassette. And then we generated 2 cell lines using the LOX, the Cree-LOX technology. So we have one allele which contains the modifications that upregulate the inclusion of the exon, and we have another allele in which, using the LOXP site that were flanking the EDA exon, then the EDA was deleted. So the first thing we did was to study development. We have seen that there was no difference in embryonic development, and this was important because I mentioned before that full deletion of the EDA gene resulted in embryonic lethality. There was, this is the extracellular matrix of mouse embryonic fibroblast, in which you can see that there is no apparent difference in the composition, except when you stain with an anti-ADA antibody, in which in this picture you cannot see because these are the fibroblasts derived from the animals which do not contain the exon. And this is the expression of the gene. You can see that in MEF, you have full inclusion in the animals which contain the mutation that upregulate optimize the splicing site, and here you don't have it. And this is the wild-type case. So then we started to analyze the RNA expression, and you can see here that in all tissues that we analyzed, so the system worked, which was important, because this is the liver, for example, in which in the normal condition you have full skipping of the exon, and in the case in which we have the homozygous mutation with the optimization of the exon, we have full inclusion of the exon. And the same happens in all the other tissues, regardless of the original rate of splicing. You can see here very clearly that in just all tissues analyzed, this is the result. So we studied wound healing, and we observed that the animals lacking the exon had abnormal wound healing. They have ulceration in the epithelium in a very high frequency, leading to problems in wound healing. We analyzed also tissue fibrosis. Fibronectin, EDA plus fibronectin participates also in tissue fibrosis, at least in vitro. This is what we knew before. Tissue fibrosis is a process in which fibroblasts or hepatic stellate cells or different cell types in the different tissues are converted into myofibroblasts, and then they start to produce high amounts of extracellular matrix proteins, and then the tissue becomes fibrotic, and this is a very important problem. And in this process, TGF-beta participates, and also in vitro, it was shown that a peptide containing the EDA plus sequence enhanced the transformation of hepatic stellate cells into myofibroblasts. So we tested this in vivo using a bleomycin fibrosis model. So we studied lung fibrosis, and we can see here that in the wild-type animal, this is collagen content, which is a measure of fibrosis. In this case, this is untreated animals, or just the mock-treated animal, and this is the bleomycin-treated animal. You can see a huge increase in the amount of collagen that is reflected here by the presence of smooth muscle actin that is a marker of myofibroblast, and then the presence of collagen that I don't know if you can see, but there are blue areas that correspond to the presence of collagen. And in the case of the animals lacking the EDA exon, then there was no increase in the presence of collagen. There was no fibrosis, extra fibrosis in those animals, and this was also related to the loss of the EDA plus animal to activate latent TGF-beta to the active form of TGF-beta. And we could confirm that this happened, and then fibronectin participates in this cycle in which the latent TGF-beta becomes active, facilitating the conversion of quiescent precursor into the activated myofibroblast, and this is also stimulated by the presence of EDA plus fibronectin in the system. So we analyzed also a large number of phenotypes. So the fibronectin participates in all this list of processes. And the idea is that we had an animal in which the only modification was the change, a very few bases that changed it, only the splicing rate of the fibronectin gene. In all cases, the fibronectin protein was present, different isoforms were present in each of the animals, but the protein was always there, and we could see that the protein affects in different ways, in different manners, all these processes that are listed here. And what was very curious for us was that the lifespan of the animal, in both cases, the animal, which is the plus plus, the one that includes the EDA exon, or the minus minus, the one that does not contain the EDA exon, in both cases was reduced. So this leads to the conclusion that, at least in the case of fibronectin, the normal regulation of alternative splicing is essential for a correct lifespan. So it means that over time, those animals are accumulating minor problems that leads to a reduced lifespan. So this is the idea. Conclusions of this part of the talk are that, well, as I mentioned before, alternative splicing is important, and the alteration of regulation of alternative splicing might produce subphenotypes. We need, this is something that I think very strongly, that we need more animals to understand the role of genes. The study of gene knockout only by doing the null mutation is not enough. It is a very important step, but it's not the only one. We can say also that, well, in most of the cases, we don't see any phenotype in the case of fibronectin unless we challenge the animal with very specific experiments, just to put the animal under stress and see if there were differences. You have to think also that animals are living in a very soft environment, so they are in the cage with they don't have any type of, let's say, how do you say, challenge to survive. So they need, in many cases, they need we need to stress what is going on. Alternative regulation is necessary for a normal lifespan. So the second model that I wanted to show you is what we are actually doing in our lab, that we developed a mouse model for the Crigler-Najjar syndrome, and we are trying to study the mechanism of disease, and also we are studying the therapeutic approaches that are possible to use in these conditions. So this model deals with bilirubin. Bilirubin is a compound that is the metabolic product of the degradation of heme. Heme is contained mainly in the red blood cells, and also myoglobin and other substances like P450 cytochromes and so on. It is converted into biliverdin by heme oxygenase. Then biliverdin is converted by biliverdin reductase into unconjugated bilirubin. And this unconjugated bilirubin, keep in mind that it's not soluble in water, so it's lipophilic. And to be eliminated from the organism is rendered soluble by another enzyme that is the Ugt1A1, which adds glucuronic acid, rendering it water soluble, and then it is eliminated normally in the bile, and then enters into the intestine, where it is converted into stercobilinogen, and part of this is reabsorbed by the intestine, and then it is eliminated as urobilinogen in the urine by the kidney. So what happens is that this step is very important. If this enzyme that is called UGT1A1 is not active, then we have a lot of this unconjugated bilirubin, and this unconjugated bilirubin is toxic. This gene is part of a very complex cluster in which you have a common set of exons plus different first exon. Each first exon confers the product different how do you say, this is the common part, and this is the specific for the different substrates. So there's a phenol cluster and a bilirubin cluster, and each one confers specificity for the different substrate. And for the case of bilirubin, this is the only one, the only gene that is capable to metabolize bilirubin. So the protein contains a transmembrane domain, and this is the variable part that gives specificity, and this is the active part of the enzyme. So different mutations have been found, and the Crigler-Najjar syndrome was divided according to the residual function of the enzyme into type 1 and type 2. Type 1 is the one that results in no functional protein, while type 2, there is some residual activity of the protein, so it is not so severe. And also there are some mutations or polymorphism in the promoter, and this polymorphism, in some cases, they are present in about 10–15% of the population, changing the levels of the enzyme and the level of bilirubin in blood. So, as I said before, Crigler-Najjar syndrome deals with bilirubin. It is a very rare disease. It affects more or less one person newborn per million. So there are not so many patients around the world. It is recessive. And then the problems I mentioned before is that we have the neonates that cannot metabolize bilirubin, so they are jaundiced. And if we don't do anything, then they develop brain damage very, very fast, and they can die of kernicterus. Kernicterus is just the specific staining of particular region of the brain, and it happens when bilirubin levels are high, and then it could be lethal, and if it is not lethal, then the survivors normally have mental retardation, just specific movement disorders, hearing loss, and deafness. And till now, there are some strategies to avoid the problems, the disease, and the most common one is phototherapy. I suppose that most of you may know that in some cases you can have as a newborn just also hyperbilirubinemia that is transient, that is because the system in the liver is still not active, so the baby has high bilirubin content, and this is non-genetic form of hyperbilirubinemia, in which it is normally treated with phototherapy. If you go to the neonatal care unit, you will see a lot of babies that are under the blue light, and then they go, after a few days, they go home. So this is not a problem. This is considered physiological and is even beneficial in some cases. But this could happen as well in this physiological hyperbilirubinemia. There are cases in which bilirubin levels are really very, very high, and the baby could have kernicterus even if the gene is active. Okay, and this is a problem that is very frequent in third world countries, and in Africa, Indonesia, it is very frequent just to have this type of problems. Babies suffering from hyperbilirubinemia, kernicterus, having even death, very frequent, and having then motorial deafness and other type of problems. As I said before, so phototherapy is the therapy normally used, and in case of peaks of bilirubin, they change the blood, they do exchange transfusions, so they take out the blood of the baby and they put fresh blood with the idea of diluting the amount of the bilirubin that is in blood. But finally, phototherapy is not efficient as such, and liver transplantation has to be done. So phototherapy is based on the conversion of bilirubin into a photoisomer that is water-soluble. So this is the wavelength that is normally used, is 450 nanometers, that is blue light. and then bilirubin is rendered soluble and eliminated in urine. So this is the type of life that the kids with this problem have to do. So they are under this type of treatment for about 12 hours per day. So you can think that the social life and the normal life they can do is very altered, and they cannot move easily from the place in which they have the lamps, so they cannot travel, they cannot move far away from their homes very easily. And the main problem is that the babies, they have the skin which is very thin, so the efficiency of phototherapy is higher. But when the kids grow up, then the skin becomes thicker. There is also a change in the surface volume ratio, and phototherapy is less efficient. Also, the kids have to increase the amount of time they are under the blue light, and in some cases they are always with very high bilirubin levels and are at risk of developing brain damage. So to try to find alternative therapies, we developed a model that we did in our lab. This model was done by Julia Bortolucci, who is here in the audience. So we introduced a point mutation, just a single base deletion in the sequence of the exon 4, and this leads to a stop codon, and the truncated protein is not active because it lacks the transmembrane domain that anchors into the membrane, which confers activity to the enzyme. So the animals, this is the strategy, that is the classical strategy of gene targeting. So we replace it, this is a targeting construct in which we replace it, this yellow, that is the mutated, bilirubin is yellow, so this is clear that it's mutated. We replace it in the allele, the wild type allele, and then by mating with a flip transgenic animal, then we eliminated the PGK neo selection cassette. And then we ended up with a mutant allele containing just a single base mutation. So you can see here clearly that the animals, which are the mutant animals, are yellow, orange, more or less due to the very high level of bilirubin. These are the wild type animals. You can see already the jaundice in the animal 1.5 to 2 days after birth, which is very convenient for genotyping the animals. And those animals, obviously, they have no protein. They have less amount of mRNA, which we haven't gone deeply into that, but we suppose that it's due to nonsense-mediated decay because of the presence of premature stop codon. And there is no activity in the liver of the animal. You can see here, this is the peak that corresponds to conjugated bilirubin in the wild-type animal. This is the heterozygous, which still contains activity, and this is the mutant, which here does not contain any peak. And what happened to the animals? Okay. So this is a Kaplan-Meier survival curve of the animals. You can see here that these are the mutant animals. The animals die more or less 50% at P5, P6, more or less. And after 10, 11 days, there is no survivor in the group of mutant animals. This is something that is also happens in babies. If you don't treat the babies, then the babies die after some time. It's not that they don't die after after 5 days as in the mice, but they die more or less before 1 year of age, 1 year and a half. And you can see here the levels of bilirubin. These are the control levels of the wild-type animals, and these are the mutants. You can see here a picture of the plasma of those animals. So this is a normal plasma, and this is the plasma from the mutant animals. So this is what you see in their plasma. The animals also have some problems. These are animals at P7. So you can see that this is the mutant animal that has motorial problems already, so cannot move easily. The legs are open just to keep balance, and it is very slow in the movement. So this animal, let's say, is committed to die in a few days. And this is what normally happens also with the babies, in the sense that they have motorial problems when they have brain damage. And well, the idea is try to avoid that. So this is just a snapshot of the brain without doing anything. This is the brain of the mutant animal, which has a different coloration of the wild type, and this is due to accumulation of bilirubin. You can also see here very clearly that this is the cerebellum, a Int'l staining. You can see that the normal features that are present in the wild-type animals, in the case of the mutant, they are reduced or even disappear. They are indicated here by the numbers. And also we analyzed just the different layers of the cerebellum. And you can see here that there is a very big difference in the how do you say, not in the shape, in the thickness of the layers, and it is resumed here. So the external jugular layer, really, it is very reduced, very less, and this is very important for the normal cerebellum function. Also, there is a loss of Purkinje cells, which are the main neurons of the cerebellum. You can see this is a wild-type animal, this is a mutant one, this is a specific coloration with calbindin to see Purkinje cells. And you can see that here the layer of Purkinje cells is very deorganized, and the cells that are remaining don't contain the dendritic tree that is seen here. And also many cells here are tunnel positive, so they are undergoing just apoptosis. They are dying. So what to do now? We did a first characterization of the animal. We know that the animal could be a good model of the human syndrome. So we decided to analyze different aspects of the disease, some of which it is not possible to do in patients directly. So one of the aspects we wanted to see is related to the presence of modifier genes. It is known in patients that in some cases babies could have very high levels of bilirubin, let's say 25 milligrams per deciliter, and after this peak they don't have any type of brain damage. And in other cases, babies that have much less concentration of bilirubin, let's say 20, 18, they suffer permanent brain damage. So there is a contradiction in this, and it is not known what is going on, which are the reasons of these differences. So everybody suspects that there are different genes that participate in modulating the penetrance of the disease. So since the mouse is a very powerful model in which we can breed and make double, triple knockouts, we selected a set of candidate genes to see if the disease was modified by the presence or absence of this gene. So we also studied phototherapy, and then we also focus in gene therapy and pharmacological therapy. This part we are starting, so I'm not going to talk about this. So the idea is to do gene editing that could be mediated, as we have seen yesterday in the talk of Wolfgang, mediated by talent. Okay. So I will start with gene therapy. So the idea is to give the animal a normal gene, and to do so we use the AAV vector that you have heard about from Mauro Giacca the day after, 2 days before. And we use a very strong promoter for that is the CMV promoter. So we injected our animals at P2, and we maintain the animals for 1 week more or less under blue light just to let the virus to express. And after that, the animals were maintained completely into normal conditions. So this is just a picture of our original phototherapy unit, in which the cages are put behind the lights. We keep control of the amount of light with a specific sensor, just to maintain always the same light conditions. And what happened? We could see that first thing, the first control was to see if the animals, which are from the C57 Black 6J background, under phototherapy, they could survive. And this is the blue light. So these are animals, mutant animals that were treated with phototherapy all the time, not only 1 week, just they are under continuous phototherapy, 12 hours a day. And even if the lifespan of the animal was extended, then the animals finally died. These are the treated animals that behave the same. The survival was the same as the wild-type animals, so we were very happy about that. And then we started to analyze so we obtained full recovery of the animals. 100% of the animals survived when they were treated with AAV expressing the wild-type gene. So we measure bilirubin levels. This is in the case of the animals treated with light. You can see that phototherapy works. So bilirubin decreases from 10, 11 to about 4, and these levels are very compatible with life. And this is the case of the treated animals. So originally we have a level of 11. After treatment, the bilirubin levels were decreased up to 2, 3 more or less, and over time there was an increase in bilirubin levels, so indicating that the efficiency of gene therapy was decreasing. You have to have in mind that we injected only once the animals that were at P4, so they were 3-4 grams more or less of weight, and then this amount of virus when diluted when the animal after animal growth. Those animals, we did the famous rotarod test we discussed over days, and we did the rotarod test, and the animals seemed to be normal, so there was no important difference against the non-treated wild-type animals. We recover as well all the problems that we have seen in the neonates in the cerebellum. So the layers of the cerebellum were just, the architecture of the cerebellum was exactly the same as in the case of the wild-type animals. You can see clearly here. And also the thickness of the layers was perfectly normal. And the number of the Purkinje cells and the presence and distribution of the dendritic arbor was exactly the same as in the case of the wild-type animals. But what happened when we analyzed the molecular part of the molecular expression of the transgene, then we have seen that in the liver there was no expression, and this was very surprising because we thought that CMV promoters should be active and very strong. And we observed expression mainly in the skeletal muscle with high levels. And when we did activity, obviously, we didn't find activity in the liver, and we found that glucuronidation activity was present only in the skeletal muscle. So this could be an advantage because for gene therapy, because the muscle is much easier to reach than the liver, so you directly can do injections much faster, much easily in different places, increasing the efficiency of the therapy. Instead, in the liver, normally it is not so simple to reach only the liver with the gene therapy. You need a kind of intervention. So we decided to see if the muscle could be a good target for gene therapy. So we used another vector, which was a vector that was driven by the alpha-1 antitrypsin promoter, which is expressed only in the liver. So you can see here the protocol of injection is essentially the same. You can see here untreated animals, and here after 2 days, the jaundice animal becomes normal, so the efficiency in the liver seems to be very high. Bilirubin levels also are low. These are the albumin levels over time. These are 17 months, so it's about 1 year and a half that the animals were treated with phototherapy. Liver histology was normal, and also rotarod was normal for those animals. We analyzed the expression, and we noticed that the expression in the liver was really much lower than in the muscle. It was about 40, 60 times less in the liver than in the muscle. And even though the amount of bilirubin in blood was comparable, so it leads to the conclusion that the liver seems to be more efficient than the muscle, even with lower amount. We doing just a semi-quantitative analysis, we could see that about 5% of activity in the liver is enough to arrive to these life-compatible levels of bilirubin. So we analyzed then what happened in the muscle, thinking also always that the muscle could be a good target for gene therapy, and we have seen that all the enzymes that produce the precursors of the substrate were present. This is RT-PCR of the liver and skeletal muscle. And we have seen also that, well, we think that this is the problem. Some of the transporters of conjugated bilirubin are missing in the muscles, so it is possible that the enzymatic reaction is slower in the muscle due to the absence of the precursor. And this is what we think. Also, if you look here, these are the levels of bilirubin directly in the tissue that were measured by Leader Trieste, that we have one of the students here in the audience. In the case of the untreated, they are very high in all tissues. When we treat with liver-specific promoter gene therapy, then there is an impressive reduction in the bilirubin contents in the different tissues. You can see the central bar. But this is not seen in the case of CMV-treated or just skeletal muscle expressing therapeutic gene. So we think that, according to this result, that we should use the liver as the main therapeutic organ for this disease. Then we studied other things. I think that we still have some time. So about modifier genes, there were some information, or this is some previous published work that indicated that some transporter of unconjugated bilirubin were present in the brain in specific zones in the basolateral side of the epithelial side of the choroid plexus, and in the case of the capillaries also in the luminal side. So this transporter could pump bilirubin out from the brain into the bloodstream, protecting the brain by decreasing bilirubin content and toxicity. So there were some experiments, one in vivo, in which was done under non-physiological conditions, in which they used MDR knockout animals to which a bolus of bilirubin was injected, and they measure bilirubin content in the brain, and they have seen that there was an increase in bilirubin content in the case of mutant animals. This was a very strong evidence, but there were also some experiments done in vitro with cells that were contradictory with this in vivo experiment. So we decided to create double knockout animals. And first thing, we changed the genetic background just to find a less severe genetic background. And we, in FBB genetic background, instead of animals dying at P5, they die at P10, P11. And then if we treated those animals, the albino animals, with phototherapy, then they all survive, while the black animals, they didn't survive. So we changed the genetic background to this one, and we did the double knockout with MRP, and we observed no difference without any type of treatment. We did the same thing with MDR, and we have seen no difference. Then we decided to try to find a less severe condition in which part of the animal could survive, to see differences in survival, minor differences in survival. So we played a bit with phototherapy just by treating the animals for 15 days, 10 days, 8 days with phototherapy. Okay, and then after this period, the animals were transferred directly to normal life conditions, and we analyzed survival of the animals. You can see here clearly these are the animals that were treated for 15 days, for 10 days, for 8 days, and these are the animals that were not treated. So we selected, in this case, this condition in which about half, more or less 50–60% of the animals survived. And then we did, these are the single UGT knockout. We did double knockout to see if, in this condition that is more borderline regarding death, the presence or not of MDR could have an effect. And we did the experiment in this condition, and we have seen, in this case, that these are the single UGT knockout animals which survive, let's say, at the beginning, about 50–60%, as we have seen in the previous graph, and then if we analyze for longer, some of them still die. But when the MDR gene is not present, so all animals die before day 25, more or less. So this clearly indicates you that the MDR is playing an important role in the detoxification of bilirubin from the brain. So we are, at this point now, we have to do other experiments just to see the histology of the animals and to see the mechanism by which this is happening. We have seen also that the animals, analysis of the animals, they have, before obviously at P16, P17, they have problems already before dying. And well, with this part, we still have to go on and to get more results. Then we are also addressed the pharmacological therapy. I will go very fast with this just to complete the talk. And this is another possibility to treat the babies, although up to now there is no efficient pharmacological therapy. So we treated we are trying to set up conditions for the treatment. So the first thing, we selected a drug that is called minocycline, that is very particular because it is already known that has some neuroprotective properties. The first thing is that minocycline, well, is an antibiotic that chelates calcium. And one important thing is that it is able to cross the blood-brain barrier. So if we have to treat the brain, the main thing is that it has to reach the cells that are suffering. So this antibiotic is capable of crossing the blood-brain barrier and has neuroprotective effect in different cell types. by reducing apparently inflammation and by reducing the amount of p38, phosphorylated p38. So it was known from another model that is a rat, that the rat is a similar model to ours in the case if we consider the mutation, but if we consider the phenotype, it's very different because those animals, the rat, which contain the same mutation, we introduce it to our animals, they have hypoplasia in the cerebellum, the same as we have. They have a loss of Purkinje cells and alteration in the different cell layers, but they don't die. So they have seen in those animals, the rat, that after treating with minocycline, they recover the normal size of the cerebellum. They recover also the number of Purkinje cells without modifying the level of plasma bilirubin in the animal. So as I mentioned before, it is acting through another mechanism different from bilirubin reduction. It's just neuroprotection. So these are we treated our animals. These are untreated animals. And when we treated with different doses of minocycline, then on the right, there are increasing doses of minocycline. And we could see here that at what we call 10x concentration, we could save about 40% of the animals. So 40% of the animals survive to the strong phenotype of bilirubin, of UGT mutation. Animals were treated up to 20 days with minocycline, and then they were living in their normal condition. To those animals, no other treatment was used, so no phototherapy, no gene therapy. And this is a very nice result because only with pharmacological treatment we could make the animals survive, at least part of them. The animals, there was a weight loss at the beginning, and then it was associated to the treatment with minocycline because this also happened to the wild-type animals. And then after we remove minocycline from the medium, then the animals recover the normal weight. So obviously we also did with those animals open field test, and the open field of the rescued animals seem to be normal, but not in all cases. In some of the cases, they have a very characteristic circling phenotype. In about 40% of the treated animals, you can see here that they stay all time in the center, just rotating there. We think that is a vestibular problem, although we still don't have any data. This is something that should be related to the combination of minocycline with bilirubin. We did also some analysis of the open field, and the group of animals that seem to be normal, they behaved the same as the control animals. Even we did the rotarod test, and you can see the first two columns. The first one is the wild-type animal. The second one corresponds to the rescued animals, the non-circling group, and at different time points, they are absolutely the same. So we conclude now that, well, we have seen that our model could be a good model for the Crigler-Najjar syndrome. There are very severe developmental abnormalities in the cerebellum. We could rescue with both extrahepatic or hepatic expression of the transgene. We could rescue all major features of the mutant phenotype. We have seen that only 5% of liver-specific UGT expression is enough to confer a level of bilirubin which are safe. We have seen also that the liver is the best target organ. And other thing, we have seen that we can modulate the disease with phototherapy. MDR transporter is an important contributor to the modulation of the severity of the disease. And we show here that minocycline is, could be a good therapeutic approach, pharmacological approach to treat at least some phases of the disease. So these are the people that did the experiment. The first part was done, well, essentially with Tito Barale when I was working with him by these people. So most of the work was done by Anil and Federico. And the UGT mice, as I mentioned before, Julia developed the model, and then also Luca, Simone, Alessia, and Fabiola are participating in analyzing different aspects. And with the help of Lorena and Mauro Giacca, who are our support for the AAV therapy, and also the different people from the Centro Studi Fegato from Trieste, from the University of Trieste, and from Prague. Okay, well, thanks for your attention.